Hi, I'm Professor Fran Boyle and I'm a medical oncologist who works in Sydney, Australia. I'm here at the Breast Cancer Trials meeting in Adelaide, and it's my pleasure to welcome Aleix Prat, who is also a medical oncologist but from Barcelona in Spain. He's also worked in the US in the laboratories associated with genomic understanding of breast cancer, and he brings together that experience of being a clinician involved in clinical trials back in Spain, but also having a very active laboratory group. And we call that, of course, translational science. So Aleix, thank you for joining us here in Adelaide. I understand this is your first trip to Australia,
yes. So
you're very welcome.
Thank you very much.
Can you tell us something about how the tumour samples and blood samples from patients in clinical trials have given us new understandings of breast cancer over the last few years?
Sure, I think this is a very important topic to talk and mention, because there is no doubt the value of clinical trials. No? The clinical trials are designed to ask a particular question and get an answer. But the value of the trials, I think, is the fact that samples, either from the tumor or the blood, can add additional information to what the trial is trying to answer. And those are very precious samples, and we've learned now, thanks to samples that have been stored for many years from all trials that have very long follow-ups, we're starting to learn that by analyzing those tumors, those bloods, we can get interesting hypotheses that then we can go and design new studies with these biomarkers incorporated into the trial. So the value is huge, and we should put a lot of effort in trying to get those samples.
And from a patient's perspective, it may be, of course, many years after the original study was done and the drug may be already approved and in use, and a laboratory such as yours goes back to those samples and learns something new. So can you tell us, in HER2-positive breast cancer, what are some of the biomarkers your group has been looking at to help us refine both our treatments and our new trials?
I think that there is no doubt that HER2-positive disease is defined by a definition, and this has helped a lot in the clinic. We're using drugs that we were not using back then, and with great benefit. But at the same time, we are starting to realize that HER2-positive disease is still biologically and clinically heterogeneous. So we need biomarkers, and I think thanks to tumor samples that have been stored in many studies now, we're starting to understand the different biology of HER2-positive disease and understand that there might be different, not only biologies, but they behave differently in terms of treatment response and survival. We have been very interested in looking at RNA and gene expression, and specifically what we call the subtypes of breast cancer. And what we and others have shown is that there is a 40% proportion of tumors with HER2-positive disease that at what we call HER2-enriched, as defined by gene expression, because you need to use gene expression to identify these tumors. And these tumors seem to be highly addicted to HER2 and benefit the most to anti-HER2-based therapies.
So Herceptin, for instance, works very well for those patients?
Exactly. And this is a very interesting observation. It does not change clinical practice today, but the fact that now within HER2-positive disease, we can identify this group of patients that benefit extraordinarily to these drugs like trastuzumab, Herceptin. Now we can design trials to try to see if we can de-escalate therapy, if we can remove some of the chemo, for example, which would be clinically very useful.
Because up until now, patients who were receiving Herceptin have to have chemotherapy with that, and they often ask the question, do I really need the chemotherapy? If this drug is as good as you say it is, do I need the chemo, and in particular, do I need as much chemotherapy? So what sorts of things might we be able to offer those patients in the future, do you think?
So I think that there might be different strategies to pursue. On one hand, maybe we don't need to give that much chemo. We just need to give a little bit of chemo with trastuzumab, for example, just a taxane for 3 months, and that could be still valuable instead of giving the full 6 months anthracycline taxanes or even a taxane with a platinum. So that's one strategy, and the other strategy is to try to identify a group of patients that maybe need even less anti-HER2 therapy. instead of 1 year less. If we can identify by using these and other biomarkers a group of patients that have good outcome and they benefit a lot from trastuzumab, maybe in the future we might give less anti-HER2 therapy, and this could be also valuable. And I think a third strategy is to try to get rid of chemo, which of course we would like to.
A big attraction.
But I think we need to be cautious about that, because HER2 positive tumors in general are highly chemosensitive, So in order to get rid of chemo, we must be very sure of what trial we would run, in what population, and feel all comfortable to do it. And I think there is some hints that maybe we will get there, but I think before we're going to get to the question of if we can remove some chemo or even less trastuzumab. I think the no chemo hypothesis or objective will be there, but I think we need a little bit more evidence.
And do you feel that the patients with hormonal HER2 positive breast cancer, so hormone positive and HER2 positive, are a distinct group as well that perhaps could be treated differently in the future?
Yes, I think that it's definitely a different subgroup of patients, the ones that express hormone receptor status. Still, there is a lot of heterogeneity with that group, but definitely, and I will show some data today and tomorrow, that even within that group, if we select the right population, potentially, yes, we can treat those patients with anti-HER2 therapy and endocrine therapy, because they already have a drug that works, which is endocrine agents. So definitely in that group is where probably the no chemo strategy could reach first.
I have now had patients who have had metastatic breast cancer who've been treated with HER2 therapy for more than a decade. And those people are extraordinary survivors, of course. Or they often ask the question, would it be possible to actually stop the Herceptin when they've been on it for such a long time without any active disease? Do you have patients of your own like that?
I do. No, not that many, because I don't see that many patients. In general, I do a lot of research, but I do have patients, and yes, some of them have been a lot of years with anti-HER2 therapy. And this question comes up, not only in patients that have been treated with several lines, but even in the first-line setting, patients that are trastuzumab, pertuzumab for a long time, and from the beginning, the pivotal trials that we included those patients are still on treatment. It's a very relevant question, and the thing is that we don't have a biomarker or at least some objective way to say we should stop. We don't have that evidence. No? I think that further studies are needed in that particular setting. Most likely some of those patients might be cured, hopefully, and this is something that probably with more follow-up we'll know. A potential, not a biomarker, but at least a platform that might be useful in that setting is ctDNA analysis, potentially.
So that's getting DNA from the tumour out of the blood, is that right?
Exactly.
Circulating
So
tumour
liquid
DNA.
biopsy is about getting the DNA in the blood without having to do a biopsy. And that just detecting ctDNA could be of value in that population, for example. I'm just hypothesizing. Of course, this needs, but if this is a patient that does not have any ctDNA in plasma, using well-validated assays, potentially there could be a group of patients that I would feel more reassured together with the other variables to make a decision like that, which is important.
Yes, because those treatments are not particularly toxic, but they do tie you down to being around a treatment centre every 3 weeks. So 10 years on, that's a big commitment, isn't it?
From a clinical point of view and for the patient, it's very important.
It's a nice problem to have, though,
Yes,
I have to say.
Exactly.
That we have metastatic patients who are those super responders. Do you think there's going to be value in studying those patients who are exceptional responders to treatment?
I think so, but it needs to be done properly, I think, because these are unique patients, and trying to put them all together in one section without having a proper control, or it's going to be difficult to find out at the end of the day what is the explanation of why those patients are responding so well. Unless we design good trials with good controls, very well define what patients are we looking at. If not, we might be lost in trying to do those analyses, getting a lot of biology, but not being able to answer the right question.
Interpret it correctly, yes. And within the Spanish clinical trials group, which is called SOLTI, what are the sorts of things that you're investigating at the moment?
So SOLTI is an academic group that runs trials in Spain and also in Portugal. 90 hospitals are attached and more than 350 members. And we are, I would say, kind of known because we try to run small studies with a lot of biology behind, so we can get back to the tumor samples and the blood and try to understand within a clinical trial who's responding and who's not. We do that in the neoadjuvant setting. We have a very strong window of opportunity trials where we give drugs for 3-4 weeks before surgery, for example, and try to understand the biology, what the drug is doing in the tumor. And also we're doing small trials in the metastatic setting. So this is our strength of this group, and what distinguishes this group from maybe other groups that run more large studies and they have big offices and more like a CRO. No, we're trying to run these small academic studies to try to open new ideas.
And that strategy you just mentioned, the so-called window trials, which involve just giving a drug for a few weeks before surgery and then measuring something in the blood or the tumour that has changed. How do patients generally accept that idea? Because it probably means delaying the surgery a little bit.
Yep. So, of course, not everybody, I think not all centers, so in our network we have highly selected centers that have run a lot of these trials. They are very used to that, and it's true. You have to explain very well to the patient what that means. For the patient might not mean much in the sense that they're not going to benefit from that drug. Potentially, it's only a short period of time that they're receiving the drug. It's true that we have to run trials with drugs that have a good safety profile. That's critical because those patients, in general, most of them are cured, and you don't want to give extra toxicity to a patient that probably with surgery and standard of care are being cured. So that's another thing. We cannot run trials like that with, let's say, drugs that have only been evaluated in phase 1 studies. But if you explain at the end of the day to the patient that the information that we're going to get out of that study is going to be very valuable, not so much for her, but for designing new trials and for other patients in the future, I think it's important because the preclinical setting, it's okay. So the preclinical setting, I mean, running drugs or testing drugs in mice or in the lab. This is good, and this has helped us design trials. But at the end of the day, unless we try to understand what the drug is doing into the tumor of patients, we won't be able probably to answer many questions, especially regarding the immune system, where definitely we don't have good models in the lab to study the immune system.
And so those sorts of studies, those window studies, I think are going to become more common for the reason that you've mentioned. And breast cancer often feels a bit like a medical emergency. I think when patients are first diagnosed, they often want to get to surgery as quickly as possible. There's a trend now, of course, to running clinical trials before surgery, isn't there? The neoadjuvant setting. And do you feel that is an area where we're going to learn also a lot more about the biology of breast cancer?
I think that one is a critical area. I think running large, or let's say huge, adjuvant trials of 4,000 or 5,000 patients being randomized to different strategies, of course we need them, but this is not probably the way to go for all hypotheses. And the neoadjuvant setting offers us a good readout to try to see if it's worth to do a 5,000 study. And definitely the neoadjuvant can offer value to the patient because we're talking about potential active drugs that might decrease the tumor size, and we know that adds value regarding the surgical outcome of that, and also regarding the prognosis of the patient. And also we have the possibility to have tumor samples before, during, and after treatment. And we already have some studies shown that having residual disease might allow us to actionate and give additional therapies after surgery that might be valuable and increase survival. So I do believe that is a critical area, and we should be doing more neoadjuvant studies, I think.
And you mentioned just before an area that's of great interest to researchers and patients, the role of the immune system in breast cancer. I've worked in the melanoma world for a while, so we're quite familiar with the fact that melanoma cells seem to be able to trigger the immune system with the help of drugs fairly easily. But in breast cancer, immune therapies do seem to need to be added to something else, don't they? Either chemo or Herceptin or perhaps an endocrine therapy. Why do you think that is, that those drugs that wake up the immune system very nicely in melanoma need a bit of a kickstart in breast cancer?
I totally agree. I think in breast cancer, I think on one hand, the good news is that there's a substantial proportion of tumors that are inflamed, so they have immune system there, so that the immune system is detecting that cancer as something that should not be there, and this is the good news.
And trying to work on it already.
And trying to work, so that's good news. It's not that these are cold tumors in general. The thing is that this is not happening, and that with just giving an anti-PD-1 drug, this is not enough. And the hypothesis might be many. I think definitely overall there must be some immunosuppressive environment going on in general that does not allow these T cells, these lymphocytes to actionate, even if we help them with an anti-PD-1 drug. So definitely combinations are probably where we need to go. In triple-negative disease, we already know that anti-PD-1 with chemo is probably the strategy, although it's true. The results are not the results probably of melanoma in general or other cancer types. So we still need to understand more of what's going on. But the good news, I think, is that there is immune system there activated, trying to do something. We just need to help them. How? Is probably where we need combinations, trials, maybe virus, other strategies that might be of value, and hopefully will shed light. It's true that breast cancer is different diseases, so potentially the strategy for triple negative might be different from HER2 positive and luminal, but we need to find out.
And I think that's a thing of interest to patients is how to enhance the immune system, and often it's a surprise to them that chemotherapy is a pretty good way to activate the immune system, because cracking open cells allows the T cells to realise that there's something happening and to wake them up. And it seems like Herceptin too probably triggers the immune system. And maybe that's what explains some of those long-term survivors of people who've done exceptionally well. Perhaps it's the fact that their own immune system has been kicked into action by the treatment that they received. We also have in Australia available new drugs in metastatic breast cancer, the CDK inhibitors, and fortunately they've all been now funded for use in patients. Do you feel like that's been a very significant step forward in treating metastatic breast cancer?
I think so. I think definitely the drugs from a safety profile, they meet the requirements, the minimum requirements we would ask. No, they're safe in general, and the benefit, the magnitude of the benefit is substantial, I think, and it delays chemotherapy use. So definitely I see the value of these drugs. Of course, still, I think we need to answer some questions regarding if everybody needs to receive this drug to start with, if there is a population of patients that maybe just endocrine therapy would do fine, and we can delay CDK4 and 6 inhibitors at second line or later stages. And the other thing is something that we're seeing now that we're using these drugs in clinical practice, that there's some patients do not benefit at all or not much. They progress at the first month's evaluation. So we really need to understand why is that and what we can offer to those patients. So the value is there, but I think we need to look more into the detail now.
Because there are still, and always were, those people who did very well with just a very simple aromatase inhibitor by itself, without the additional monitoring blood tests and so forth that you need for using a CDK inhibitor.
From a patient perspective, that would be wonderful, no? If you could have a biomarker that says, no, you don't need the CDK, we can wait, and you can be treated with endocrine therapy from a quality perspective point of view, I think it would be good, but we need a biomarker. We have presented some data, and I will present some data tomorrow regarding a prognostic biomarker in this setting, trying to combine gene expression together with clinical variables in a single model to try to identify patients with better outcome versus worse. And this is something that we presented at ESMO Breast, and hopefully we're trying to validate in the pivotal phase 3 trials, Mona Lisa especially. The question is if that's going to be good enough. If predicting prognosis or survival in early disease is clinically valuable regarding the use of chemo, yes or no. In the metastatic setting, how much this will add to what we're doing, we'll see. But I think we need also prognostic biomarkers, so we can select this group of patients that we can treat with endocrine therapy on its own.
And in choosing treatment for patients with metastatic disease, we're also starting to realise that potentially rebiopsying one of the metastases, or maybe the liquid biopsies that you mentioned before, taking some blood and identifying something in the blood that helps us to find mutations or targeting strategies. That's tough though from a patient's perspective to have to think about a biopsy again. Do you think that's going to become increasingly important?
I think so. I don't think the tumor evolves naturally and evolves by trying to become resistant to therapy. So either we study that, or it's going to be, I think, quite difficult to try to improve outcomes in the metastatic setting. Unless we study those tumors upon progression or during therapy in that setting, it's going to be very hard to design trials that provide meaningful outcomes for the patient. So I understand it's biopsying the liver, the lung, the bone. It's for the patient perspective, it's definitely tough. And I also think that liquid biopsy is not going to provide us all the answers. DNA is important in breast cancer, but not probably like lung cancer. No? When you detect a mutation, you have a drug, 70% response rate. I think we still need to go to the tumor itself and try to understand more than just DNA. But again, I think this needs to be done, especially within the trials. Again, going back into our first conversations, no? I think integrating these biopsies within trials so we can test hypotheses or what's the value of that biology, it's going to be hard to move the field forward.
And do you think we can learn better how to assess what the immune system is doing within that tumour by looking for biomarkers of immune activation, or whether the tumour is inflamed or not?
So I think that the immune system, definitely analyzing the tumor sample is going to be important, because there is where the immune system is and trying to do something. But at the same time, probably the blood will also allow us to study the immune system and as a surrogate biomarker of what's happening in the tumor. And there's already some data, especially in melanoma, trying to identify a specific population of lymphocytes in the blood might tell us how these lymphocytes are in the tumor. So I think the immune system is definitely critical, but potentially also the blood might be of value in that regard. But I think we're a little bit lost still of how to analyze those samples.
It's a patient's question that comes up all the time. Patients with metastatic disease sometimes are cured. It's not so common, and we probably keep that information a little bit to ourselves because one doesn't wish to offer false hope. But do you think in our working lifetime metastatic breast cancer will become a curable disease?
I think so, but again, with caution. In a proportion of patients, a small proportion of patients, curation, I think it's an objective, and I think we can say it properly. Although still we need more data, more results, and potentially more drugs. And the other thing is to make it more chronic, to make a chronic disease and to live with metastatic disease. And I think that's also, a more realistic objective, to see those patients that live with metastatic disease, that you can see in the CT scan, but they can live long periods of time having almost normal life. I think that's the more realistic objective. The curation, I think it's also an objective with a small proportion. Hopefully it will increase as we understand more about the biology, as we get more drugs approved, and definitely these are good news in general for the metastatic setting.
What do you think are the barriers to designing good clinical trials today?
The main one is funding, I think, because ideas, I think they are very good ideas. I keep hearing very good trial designs from many collaborators. The problem is how to implement that trial. Trials are expensive in general, and I don't know here in Australia, but at least in our country, in Europe, governments do not invest in running clinical trials, academic clinical trials, and it's making us very difficult. As an academic group, funds at the end of the day come from pharma, which is great. They provide funds to run our own academic groups studies, but we have limitations on that. So I think funding is one of the limitations. The other limitation is trying to have more collaborations abroad. I don't think, as we are subdividing breast cancer into smaller proportion of patients, small groups with that alteration, we need to do this in collaboration. I don't think single groups can do it by themselves, or they would take a long periods of time. And I think collaboration is important, so trying to find ways to collaborate with other countries, with other groups, could be important. And I say that because I think we are willing to collaborate, but at the end of the day, when you put this into practice, you have the different laws and you have different issues that at the end of the day make it very difficult to collaborate. So I think collaborating is definitely something we need to pursue.
So it's very good that Spain and Australia are only 8 or 10 hours apart for teleconferencing. But once you add Boston into that mix, somebody's awake at midnight. So one of the concerns, of course, for Australians is that it's usually us who are awake at midnight if we want to project our ideas to the world. So maybe projecting them to Spain is not quite such a long bridge. You mentioned before about doing the laboratory work, the analysis of the RNA and the DNA in the tumours. And it seems to me that that's an area that's often hard to fund as well, isn't it? Because that involves PhD students, laboratories, equipment, etc. And so that's one of the advantages, of course, of having funding from the public, from donations to support a trials group. That allows to add that extra layer where the gold, if you like, is to be found. The pharma perhaps might supply the drugs, they might supply some money to actually make the trial run, but to do that laboratory work is really where having some extra money I think is extraordinarily valuable, and one of the things that we try to engage the community in here. So do you have in Spain the support of the local community for your research?
So the good news is for breast cancer, usually the community is quite engaged in trying to raise funding, and there are many groups in Spain. Actually, there is one very strong group dedicated to metastatic breast cancer patients. It's called the Spanish Metastatic Breast Cancer Patient Group. It's a foundation, and they have raised a lot of money in Spain to then have grants that then can fund analysis of my lab or others, and I think that's a way to get funding from the community. And at least for breast cancer, we have that. Other cancer types, it's much more difficult to get the community involved, but in breast cancer, definitely. And we have examples in the US with huge crowdfunding, almost companies, no? Like Susan and Komen Foundation. They raise millions and millions, and then they have amazing grants that they're giving. So I think also this in Spain is picking up. I think we're a little bit behind other countries, but it's definitely very important.
Well, thank you very much for talking with me today, Alex, and you've really raised a number of issues that are of great importance to those of us who are involved in breast cancer research. The absolutely critical importance of tissue and tumour samples, the critical importance of funding to do that translational work in the lab, and some new strategies for designing clinical trials to try and speed up progress. And the hope, of course, that in a few years' time we'll be back here talking about our cured patients with metastatic breast cancer. So thanks very much for making the trip to Australia. And we hope that discussion about collaboration will be part of what happens while you're here.
Perfect. Thank you very much.